students. So today is our second lesson in cell biology. In the previous lesson, we had covered the cell, the unit of life. For those who have missed it, the video link is provided in the description of this video. Today we will be starting the biomolecules, and in subsequent lessons we also cover the cell cycle and cell division. So the objective of today's class is we will learn about carbohydrates and lipids, but the biomolecules includes proteins and nucleic acids as well, so which we'll cover in the next subsequent lessons. But today's class we will cover only the carbohydrates and the lipids. And among carbohydrates and lipids, we will cover the basic structure, functions, building blocks, along with examples. So let's start. For example, a friend of yours says that I have been eating carbohydrates all day. So what food comes into your mind? What would you assume that she is eating? So you may be thinking that it may be processed food like potato chips, or it may be chocolates, or candies, or bread or croissant, or it may be spaghetti or noodles, or it may be as well as rice. So all these are sources of carbohydrate. You may assume this. But it's very important to understand that the carbohydrates are found not just in grains or in sweets or processed foods, but every food group. For example, fruits. Fruits, we have carbohydrates. In other foods like grains, every grains, carbohydrates are present. And even the cheese, butter, the carbohydrates are present in quantities. So we can conclude that carbohydrates are the most abundant nutrient, except water, in the diets of most humans around the world. So let's learn more deeply on carbohydrates. So as told before that it is the most abundant biomolecule on the earth, and the main source of energy. Carbohydrates are the source of energy for every living organism, let it be plants or animals. So some structural components of carbohydrates are cell wall, where it is made up of cellulose. So cellulose is a carbohydrate. And also the nucleic acids, that is DNA and RNA, they have sugars inside it. And the name of the sugar is ribose. So we can see that how abundant is it. And the saccharide word we will use more in this topic. So it's nothing but it is a sugar. So if it is another name of sugar is saccharide. Now the building blocks of carbohydrates are mainly carbon, hydrogen and oxygen. So carbon, hydrogen and oxygen contains for example glucose, and glucose is an example of carbohydrate. But we have to see some proportions. All the carbon, hydrogen and oxygen are present in a particular proportion, and the hydrogen and the oxygen especially are in the ratio of 2:1. So general formula of carbohydrates is CnH2ON or H2OY. For simple sugars, the N and the Y value is same. But for complex or complex sugars or polysaccharides, the N and the Y value is different. But you have to remember the general formula is like that, where you can see that hydrogen and oxygen are in the ratio of 2:1. So let's see examples here. So in this case, glucose is a simple molecule, simplest sugar molecule, where N is equal to 6 and M is also equal to 6. And you can see that H and O are in proportion of 2:1. Okay. And so the general formula, molecular formula of glucose is C6H12O6. For sucrose, which is a disaccharide, so it has N is equal to 12. And M is equal to 11. So you can see here the N and M value is different, but there is always the hydrogen and oxygen are in the ratio of 2:1 in case of carbohydrate. Okay, so the general formula of sucrose becomes C12H22O11. Now, carbohydrates, the types if we see, they are mainly simple carbohydrates or complex carbohydrates. In simple carbohydrates, it can be further categorized to monosaccharides and disaccharides. As I told you before, that saccharides means sugar molecules. So it's one sugar molecule, and disaccharides means only two sugar molecules. Let's see examples. So for monosaccharides, example can be glucose, fructose, and these are just structures of glucose and fructose. For disaccharides, we can take examples as told two sugar molecules. So for example, sucrose. Sucrose is made up of two sugar molecules, two monosaccharides. So one is glucose and another is fructose. Similarly, the galactose is made up of two monosaccharides, which is glucose and galactose. So the complex sugar molecules, also called polysaccharides, are made up of many sugar molecules. It is more than two sugar molecules per unit. And the examples are starch, glycogen, fibers. So here you can see it is more than 2 units. So this is 1, 2, and 3, 4, but there are many. And so we can understand the process of forming a disaccharide molecule from a monosaccharide molecule. So let's take a monosaccharide molecule. The simplest one is glucose. So you can see the molecular formula of glucose is C6H12O6, but structurally it looks like this. So we are taking two monosaccharide molecule, and importantly I want to highlight these two hydroxyl groups. So OH is a hydroxyl group. So we are highlighting these two hydroxyl group of two different monosaccharide molecules. So what will happen if I take out one hydroxyl group and the hydrogen of another hydroxyl group? So if you can see that one hydroxyl group and hydrogen becomes water, that is H2O. So it becomes a water, and I remove it, I pull it out. So what will happen? So you can see here only one oxygen was left, and this bond, whatever is formed here after removal of one water molecule, we call it as glycosidic linkage. So this is very important formation. So glycosidic linkage is formed after removal of one water molecule from the two monosaccharide molecule. So after that they join together and form a disaccharide. Okay, so this is the basis of formation of not only disaccharide but every polysaccharide in sugar molecules in carbohydrates. So we can see here in detail that the two sugar molecules and monosaccharides forms a disaccharide by pulling the water molecule. So this linkage we call it as glycosidic linkage. So to conclude, we pull out one water molecule to build a bond, and the name of the bond is glycosidic bond. Okay, glycosidic bond. Now can you tell me what type of reaction is it? Because we are pulling out one water molecule. So removal of water molecule. Yes, you are right. It is dehydration. So dehydration, when D means removal, hydration means water. So removal of water. So dehydration synthesis. Why synthesis? Because we are forming a glycosidic bond. The second name or another name of this reaction, where we pull out one water molecule to form glycosidic linkage, is also condensation reaction. So either any one name is fine, dehydration synthesis or condensation reaction. I have taken a disaccharide, which is formed again by removal of one water molecule, and I'm adding a third monosaccharide. So the hydroxyl group is here, and they form another glycosidic linkage. So this is the way, and this is also formed by removal of a second water molecule. So based on the number of glycosidic linkage, you can guess that how many water molecules are removed. So this way it can form polymers, just like starch, glycogen, cellulose, etc. Okay, and the basis is the dehydration synthesis reaction or condensation reaction. Okay. So these are just examples. So two glucose molecules, these are monosaccharides. On condensation, they form maltose. Okay, and this is the glycosidic linkage and one water molecule. Similarly, glucose plus galactose forms lactose and one water molecule. And glucose plus fructose on condensation, that is, condensation means removal of one water molecule, they form sucrose. So what is the opposite of removal of water molecule? What will happen if we add water molecule? Yes, you are right. So if we add water molecule to a disaccharide, it will break down to two monosaccharides. Here is an example. So if what will happen if we add water molecule, so you can see here the water molecule means one hydroxyl and one hydrogen atom. So it will form the respective monosaccharides. So a reaction where a water molecule is added. So here we add a water molecule to split up a complex molecule. So this one was a complex molecule and we split it up. We call it as hydrolysis. Hydrolysis means breaking down and hydro means with the help of water. So we are breaking down a complex molecule with the help of water. So this reaction is called hydrolysis reaction. Structure of a glucose molecule, and my question towards you is that, is the glucose soluble in water? Yes, it is soluble in water. But why? So if you see here, glucose has some hydroxyl groups or OH groups, and these OH groups are the reason for its solubility in water. So we can see why that hydroxyl groups are soluble or help in solubility, because they are polar in nature. So the hydroxyl groups are polar and hence the glucose is soluble in water. So let's see some more examples of hydrolysis. Like if you take disaccharides of maltose, so with the help of maltase enzyme, you can break down two glucose molecules. Hydrolysis, again to remind you that it is breaking a disaccharide or breaking a complex sugar molecule with the help of water. So hydro means water and lysis means breaking down. So here it is also facilitated by enzyme in our body, which is present in small intestine. For lactose, it's an example of which breaks down to glucose and galactose in presence of lactase. And similarly, the sucrose breaks down to glucose and fructose in presence of sucrase. Now let's dig deep into polysaccharides, which are the starch, glycogen and cellulose. So coming to starch, starch is the most important source of carbohydrates in our food. Okay, so like bread, chapatis, rice, etc. It's rich in vegetable foods. So it's most importantly the cereals or potatoes are examples. Very importantly, it is not found in animals. It's only and only found in plants. So, and finally, it is detected by an iodine test. So, let's come to the structure of starch. Starch is a mixture, as you can see here, of a straight as well as branched chain. So, it has a straight chain as well as the branched chain. And they are formed by multiple condensation because they have multiple glycosidic bond. So condensation is removal of water molecules, right? And the hydrolysis of the starch, which is the opposite of condensation, which is done with the help of water molecules to break the glycosidic bonds, and they result in releasing glucose chains and finally to glucose molecules. So many glucose molecules forms the starch. Okay. So let's see the hydrolysis of starch in details here. So this is a starch molecule and these are the glycosidic bonds, and it takes place with the help of the enzymes. As I told before, that amylase is an enzyme which is found in our mouth, and which degrades the starch while chewing the food into the mouth to maltose. So starch is converted to maltose with the help of amylase in the mouth. Okay, this amylase is present in saliva. The second important thing is that the maltose is further converted to glucose with the help of maltase, and maltase is present in the small intestine. So it's very important to note that the starch digestion does not take place in the stomach or abdomen. It takes place either in the mouth, where it's converted to maltose, and then it is further converted to glucose in the small intestine. So, for example, this is your digestive system, and you start the starch chewing in the mouth, where the amylase enzyme is here, and it's converted to further glucose chains. Okay, and then further glucose chains, maltose is there, and maltase enzyme is present in the small intestine. This is the small intestine, and here maltose converts the gets converted to glucose with the help of maltase enzyme. Okay, now let's see in big zooming. Here, for example, the glucose molecules are there. Okay, and here it is the intestinal cells. So here it is your bloodstream. So the glucose is gets permeable to the bloodstream. Okay, so the final digestion of the starch takes place in the small intestine. Now that's for carbohydrate. Just to have a revision that if you take iodine solution uh, and which is this color, orangey brown in color, and you drop it on starch food containing. So if the food contains starch, then the color of the iodine changes to blue black. So if the blue black color you get it, then the starch is present, else the starch is not present. Now coming to the second polysaccharide that we'll discuss is glycogen. So glycogen is importantly an animal starch. It is not present in plants, and it is formed when numerous glucose units are condensed to form highly branched chains of glucose units. You can see here it is branched chain of glucose molecules present in animals, not in plants. And it is a storage form of carbohydrate, just like the starch. It is also a storage form of carbohydrate, and it is also found in fungi as well as animals. And for animals, the form it is generally stored in livers and muscles. Now, if you see here, for example, this is a liver, and the formation of glucose to the glycogen. Okay, so many glucose molecules polymerizes to form glycogen, and glycogen is stored in the liver. So this process is called glycogenesis. Glycogenesis means it is forming glycogen from glucose molecules. And whenever we need, for example, carbohydrates, for example, we fast sometimes, and at that time, the liver, the glycogen, whatever is there, it degrades or decomposes to glucose molecule. And that process we call it as glycogenolysis, because it's breaking down of glycogen to glucose molecules. So one is glycogenesis, where glycogen is formed, and glycogenolysis means glycogen is degraded. Now a question to you is that why starch and glycogen are used as storage materials? Because these two polysaccharides are storage polysaccharides. Okay, but what property makes them unique? Let's see. So here, if you see here, these two are the structures of starch and glycogen. So first important property that both of them have is that they are insoluble in water, and hence they do not change the osmotic pressure of the cell. If, for example, a molecule changes the osmotic pressure, that means the size of the cell changes, and hence it is difficult for them to maintain it as a storage molecule. So it is insoluble in water. This is the first important property. The second important property, they are very large molecules and hence unable to diffuse through the cell membrane. So it is a second important property that makes them a storage polysaccharide. The third important property is that it can easily be hydrolyzed For example, whenever we need glucose, whenever we need energy, the process of hydrolysis takes place very easily to both starch and glycogen. And the final and important property is that it is very compact, so it occupies a very less space. So these four properties makes the starch and the glycogen a unique storage polysaccharide. Very importantly to remember them, the mnemonics. So you can see here L for large molecule, I for insoluble, C for compact molecule, and E for easily hydrolyzed. So this makes your these properties makes the starch and glycogen a storage polysaccharide. Coming finally to the cellulose. We know that cellulose are the carbohydrates present in the cell walls of the plants. Okay, and most importantly, it is different from glycogen and starch. So, cellulose are actually formed by the hydrolysis of beta glucose rather than alpha glucose. Let me give you a brief introduction on the glucose. Glucose, we know that the molecular formula is C6H12O6. Okay, so here no doubt about that. But there are different orientations of hydroxyl groups, and hence based on the orientation we name one of them as alpha and another name is beta. So it's good to remember that both this starch and cellulose have glucose, okay, but the orientation of the glucose molecules is different. For example, starch is made up of alpha glucose and the cellulose is made up of beta glucose. Okay, so cellulose are formed by the hydrolysis of beta glucose molecules. So this is the basic structure of cellulose molecules, and it's important that why the cellulose is so strong. So the cellulose is strong because they have straight unbranched structure. So if you look here, this is the cell wall. We zoom in further and we get the cellulose fibril, and each cellular fibril have a microfibril, and each microfibril have many chains of these cellulose unbranched chains, and each unbranched chain have hydrogen bonds between them. Though these hydrogen bonds are weak, generally hydrogen bonds are not so strong. But this cross-linking, many hydrogen bonds, make them a very strong unit. And hence what happens, the cellulose is very strong. Human body doesn't have the cellulase enzyme which can digest the cellulose, but yes, many animals have the cellulase enzyme who can digest, for example, cows, buffaloes, etc. Okay. So to conclude the functions of carbohydrates, as it's a good energy source, for example, glucose, lactose, galactose. So if you see the O's, OAC suffix, then you can think about that, yes, it's a sugar. Most important function of sugar is they are energy providers, like the starch, glycogen, which are as storage polysaccharides. And also it is a structural polysaccharide just like cellulose. So the distinguish between starch, glycogen versus cellulose is that cellulose is structural polysaccharide, whereas starch and glycogen are storage polysaccharides. The starch is found in plants, glycogen in animals. Starch and glycogen are insoluble, compact and easily hydrolyzed. Use the mnemonics LICE, L-I-C-E, to understand and remind yourself about the properties, unique properties that makes starch and glycogen easily a storage polysaccharide. Cellulose is a structural polysaccharide made up of glucose monomers, which are beta glucose, right? And cellulose forms microfibrils, which are strong due to many hydrogen bonds. Now coming to lipids. So lipids are non-polar organic molecules, and the non-polar is the word non-polar means it's not polar, unlike the glucose molecules, right? So and they are hydrophobic. Hydrophobic means they are not water loving, and the reason the diverse means it's not as simple as carbohydrates, and there are various types of lipid molecules. Now, functions and examples. Lipids are good place to store energy. For example, the adipose tissues store energy as a compact form. Their structural material, especially phospholipids and cholesterol, are found in abundant in all cell membrane. We can use them as water barriers. Because of their non-polar hydrophobic nature, they make a good water barrier. Just oils and waxes are examples for that. And another function of lipids are that they are messenger molecules. For example, many hormones and steroids are composed of lipids. Functions on fats are that they provide a good insulation. Also, they provide the cushion for vital organs like heart and liver, and also they transport fat-soluble vitamins. Last but not the least, they also provide the essential fatty acids. So, in food, the fats enhances taste, flavor, aroma, crispiness, juiciness, tenderness, smooth texture, creamy feeling, and fats makes us feel full that we have eaten something. Also, the triglycerides in the food, we can find triglycerides in fruits and vegetables, breads. Avocado contains is a fruit where the high quantity of fat is present. Also, bread, cereals, rice, pasta, grains has some quantity of fats. Meat, poultries, dairy foods, fats, oils, and condiments are examples for that. Now, the building block of fats is fatty acid. Okay, let's understand the fatty acids in a bit more detail. So, fatty acids, the basic structure of fatty acids is that if you see that I'm making a hydrocarbon chain. So, hydrocarbon means a chain containing carbon and hydrogen. And if you see here, each carbon has 4 hands or 4 valences. So, it is just a chain of hydrocarbon. And here, after forming a chain of hydrocarbon, on one side it is I'm fulfilling it with hydrogen, so it forms a methyl group, and on the other side it's carboxylic acid. Why? Because this is a fatty acid. Acid means the functional group has to be carboxylic group. Okay, and this end we call it as alpha end carboxylic group. And the other part, which is we call it as omega end fat methyl group. So it's a methyl group, that is CH3, C and H3. This group we call it as methyl. And so fatty acid is nothing but it is a chain of hydrocarbon and along with a carboxylic group. So this is a carboxylic group, C double bond OOH or C double OOH, and the methyl group is on the other hand, and there is a hydrocarbon chain in between. Now, there are many types of fatty acid. So, types of fatty acid, basic fatty acid structures types are saturated fatty acid, and another is the unsaturated fatty acid. Saturated means what? It is satisfied, means it does not want to take any other atoms. It is fulfilled, it feels fulfilled. For example, the saturated fatty acid is present in butter, in cheese, in ghee, etc. And unsaturated fatty acid is present in sunflower oil, olive oil, even the fishes. So they are examples of unsaturated fatty acid. Now, if you dig deep into the saturated fatty acids, this is a chain of saturated fatty acid. And here you will see that the main chain, whatever is that, the main chain doesn't have double bond. So no double bond is there in between the main chain. Of course, there is a carboxylic acid group on one side and methyl group on the other side. But whatever main chain is there, there is no double bond. So in this way, we can distinguish which one is a saturated fatty acid. On the other hand, the unsaturated fatty acid has a double bond, one or more. So if it has a double bond, it is unsaturated. So we will just remember this for now. And if you see there are more than one double bond, then it is called polyunsaturated fatty acid. For example, if this is a fatty acid and you can identify here that is there is more than one double bond. Now, how can we build a triglyceride? So triglycerides are made up of one glycerol molecule. So this is a glycerol. So all means hydroxyl group is there. Okay. So there are three hydroxyl groups is there. So it is one glycerol molecule, and along with it we take a fatty acid. So if you see here, this is the fatty acid, the long chain. Hydrocarbon chain is there, and this is the carboxylic acid group, so it becomes a fatty acid. So I'm taking one glycerol molecule, one fatty acid, I take a second fatty acid, okay, and this is the same fatty acid, same or different, but I'm taking it's another fatty acid, and I'm taking a third fatty acid, and hence the name is triglyceride. So triglyceride, why the name is triglyceride? So glyceride comes from the molecule glycerol, and tri from the three fatty acids. Okay, and just to reinforce the fact that the number of hydrocarbons, whatever is present in between, they may vary. It doesn't matter. The main reaction happens with the carboxylic group, which is the C double bond O OH. So this functional group participates in the main reaction. So if you see here, the one hydroxyl group of the glycerol molecule and the hydrogen of the carboxylic functional group forms a water molecule. So for example, here OH and H becomes H2O. Another OH and H becomes H2O, and finally here also. So you can see here that we can remove three water molecules from this, and this reaction is the condensation reaction, because we are removing the water molecules. And as a result, after removal of the three water molecules, we get this product where the bond is the glycosidic bond. Okay, so glycosidic bond means as a bond which is formed due to the condensation reaction. So this is one glycosidic bond, this one is the second glycosidic bond, and this one is the third glycosidic bond. Okay, and these are the three fatty acids. So this is just to remind you, and you will not forget for the future, it is a glycosidic bond formed due to from the condensation reaction. Okay, and this is a basic structure how you can build the triglycerides. You can change the number of carbon atoms in between. It may vary, but whatever reaction takes place, we have to understand that glycerol is fixed. So there is one glycerol molecule and there are three fatty acids. Okay, so one glycerol and three fatty acids, they form what? They form triglyceride after removal of three molecules of water. And what type of reaction is it? It is condensation reaction. Okay. Similarly, we can think of the opposite thing. There is one molecule of triglycerides. If I add three molecules of water, then it will form one glycerol plus three fatty acids. It's just the opposite. And yes, remember the reaction name? It is the hydrolysis reaction, because we are breaking down a complex molecule. Here it is triglycerides with the help of water. Now, phospholipid is another category of lipids. It's also lipids, and it is present in the cell membrane of every cells. And it has a phosphate head and a lipid tail. So this is the structure of phospholipid. If you see here, this one is the phosphate part, this one is the glycerol, and this is any fatty acid. Okay, so here this part, phosphate part, is hydrophilic, that is water loving, and this part, that is the fatty acid part, is called the hydrophobic. So if you remember, in the last class we had covered the fluid mosaic model, and the reason for maintaining the fluidity of the cell membrane is because it has a both a hydrophilic head and a hydrophobic tail. Okay, and you can understand the structure. Sterols are also another example of lipids, where you can see that it is unlike the phospholipids or the triglycerides. They have ring-like structure. Okay, they are non-polar. So if there are 4 ring structure, you can understand that yes, it is also a lipid. And examples of these sterols are sex hormones like testosterone, estrogen, etc., and cholesterol. Cholesterol is also an example of sterols. Okay. In the coming biology class, we will cover the other 2 biomolecules, which are proteins and nucleic acids. But before proceeding further, you please check your understanding through some of the questions based on carbohydrates and lipids. Stay tuned. Thank you for watching.